Hi there, my name is Ryan Dickerson. I'm a student in the Brady School of Medicine Class 2025, and this is our video on embryonic development of the cardiovascular system. As always, here's our mission statement for Scrubs. Scrubs is a student-driven initiative that aims to develop supplemental resources for current and future cohorts that will pass through Brady. These are made by students who have gone through the course and are developing resources that we wish we would be exposed to during our time in the course to aid in our learning. As always, brief disclaimer, these resources are made by students for students, are not made by faculty. As such, they are a supplement to your course material, and they're no way a replacement for the material presented by your faculty. With that, let's go ahead and dive into the material. Okay, so in order to understand a little bit about the development of heart, I think it's important to recall a little bit of adult anatomy. And I apologize in advance, this video is going to be a little bit longer than our normal standards. So in thinking about adult anatomy, it's important to remember that we have an inferior vena cava, superior vena cava journey into the right atrium that is carrying deoxygenated blood from the upper limbs and the lower limbs. The right atrium will then transmit blood into the right ventricle that goes through the tricuspid valve. As we're in the right ventricle, it is then going to be pumped into the pulmonary trunk, and there goes our pulmonary valve there. And the pulmonary trunk is going to bifurcate, going to the right and left lungs. Okay, that is carrying deoxygenated blood. Now, coming back to the heart, we have oxygenated blood coming through our pulmonary veins, which we can see in the back. Those are draining into the left atrium. Left atrium is then going to transmit blood into the left ventricle, right? And that's happening here at our bicuspid valve or mitral valve. That left ventricle is then going to pump blood into our aorta, and we can see that the aorta is going to arch and come out on the right side of the heart in relation to the pulmonary trunk. This will be important, as we'll see later. And then the arch of the aorta is going to go over, and then it's going to descend into the thorax and abdomen. All right, and then we have these great vessels coming off. This is our branches, brachiocephalic, and then we have our common carotid as well as our subclavian on the left. Okay, looking from an external view of the heart, a couple other structures we can see. Again, really want to point out these great vessels as they come in and out of the heart. Superior vena cava and inferior vena cava journeying into the right atrium. We have our ventricles down here in the front. We see our pulmonary trunk, which is carrying that deoxygenated blood once again to the lungs. That is coming from the right ventricle. And then we have our aorta, and this is the arch of the aorta as it's coming around. That is again going from the left ventricle, wrapping around, and then it's going to descend down into the thorax and abdomen. We can also see our pulmonary veins here draining into the left ventricle, left atrium. And remember that the left atrium is situated in the posterior aspect of the heart. It's going to be the most posterior structure in anatomical position. All right, one other thing to point out briefly is remember that we do have coronary artery and veins. That's going to be supplying the heart or the ventricles with blood. The main one here, we have our descending or anterior interventricular artery. Okay. And then over in the back, we're going to have our marginal, right marginal specifically, because here's our right CA going off. Again, we'll talk a lot more about this in the anatomy video, but this is just a brief overview so that we know what our end result is for embryonic development. So going back into the third, fourth, fifth week, this is really when we start to have our organs develop. The heart is one of the first organs to develop, so it'll really be occurring during the third and fourth week. But again, screenshot in time, we're starting to have our body folds occur, we're starting to develop cavities, and now we're going to start to have organs develop. So this is kind of where we're at embryonically. Okay. One thing I want to bring our attention back to is remember around this time is where we're forming those body cavities and the head fold's coming down. That head fold is pushing down to bring the intraembryonic colon into different regions. And we see here that it's going to be situated directly in front of the developing heart. And this region is going to be our pericardial cavity. And just like in the adult, we know the pericardial cavity is going to surround the heart. So this again is showing us that relationship occurring embryonically. Now, kind of previewing how the heart is going to develop, I'd like to take a large, general scale, and then we're going to dive into details. So initially, we're going to have the heart tube forming, and it's going to be a single tube that's going to eventually develop into the future heart. In order to do so, you're going to form those primitive chambers. So that's going to be forming the atrium, the ventricles, and the chambers that we recognize in the adult. Now, what we're going to see is these are eventually going to form in a superior to inferior direction, which is not how we see it in the adult. So we need to position those chambers in different ways so that we get the adult structures. Now we know we have a right ventricle, right atrium, left atrium in the adult, but initially in development we're just going to have atrium and ventricles. They're not going to be partitioned, so we're going to need to partition those into a right and left side so that way we get the normal blood flow that we would expect in adults. And lastly, we're going to have remodeling of chambers. This is just going to be changes from the fetal stage into the embryonic stage, the embryonic stage into the neonate stage, which is going to allow for the blood flow transition from maternally derived to fetally derived. Okay, So now thinking about the formation of the heart tube, there's a couple of steps that we're going to start with. So initially, the developing heart is going to arise from the cardiogenic area, and its first step is going to be giving off angioblastic cords. So kind of thinking through the terminology here, angio means vessel, blastic, germ or sprout. So this is basically saying that you're forming blood vessels. Now these angioblastic cords aren't actually vessels at this point, because they're not canalized. So they haven't formed a canal to allow blood flow to transmit. So the next thing that has to happen is we have to canalize those angioblastic cords into what we'll call endocardial heart tubes. So now that we have a canal, we're going to have two endocardial heart tubes on either side of the developing embryo. But we know that the heart isn't two separate structures, it's one big organ, so we need to fuse those heart tubes together. So those endocardial tubes are going to fuse, and that fusion is then going to be referred to as the primitive heart tube. Okay, so the endocardial heart tubes fuse, and then eventually we're going to have dilations that are going to form. Okay, so once again, endocardial tubes fusing to form the primitive heart tube, and then you can see here, we still can see what used to be the two endocardial tubes coming in and leaving, but now there's fusion in the midline, which is what is going to be our developing heart. So as the heart is developing, it's going to start to give off some dilations. So the most superior dilation we're going to see, we're going to call the truncus arteriosus. Inferiorly, we're going to have the bulbus cordis, followed by the primitive ventricle, atrium, and most inferiorly, the sinus venosus. So these are all terms that are really good to know. We're going to be talking about this a lot for the rest of this video. So again, superiorly, truncus arteriosus, bulbus cordis, ventricle, atrium, and sinus venosus most inferiorly. Okay, so in that last slide, you could see that the truncus arteriosus was most superior, sinus venosus was inferior, and then everything else was kind of in a direct midline. Now what you'll notice is initially the atrium is situated inferiorly to the ventricle. But we know in the adult anatomy, the ventricles are actually situated inferior to the atrium. So something has to change somewhere in development. So what's going to happen here is the truncus arteriosus and sinus venosus are pretty much locked into position based on additional structures that they're attached to. So truncus arteriosus is attached to the pharyngeal arches, which are going to rise superiorly, and then the sinus venosus is attached to septum transversum, which, if you recall, is going to develop components of the diaphragm. So that's attaching the sinus venosus inferiorly. That said, there's going to be differential growth of the ventricle and bulbus cordis, which is going to allow for rotation. And this rotation that we're going to call is the bulboventricular loop. And the end result of the bulboventricular loop is to have migration of the chambers into their final positions that we would recognize in adult anatomy. So what's going to happen is the bulbus cordis is going to move to the right while the ventricle moves to the left. So what you'll see here is the ventricle here is shifting to the left, whereas the bulbus cordis is shifting to the right. At the same time, the atrium and sinus venosus are going to be pulled posteriorly and superiorly. So again here, the ventricle shifting to the right, shifting to the left, bulbus cordis shifting to the right. You see the atrium is getting pushed backwards and superiorly, and then eventually we're going to have the structure here, which is much more similar to what we have in the adult anatomy. We have the ventricle in the inferior left pole. We have the atrium, which is now posterior behind the ventricles. We see that the bulbus cordis has moved over towards the right. And then again, the truncus arteriosus is locked into place superiorly, and kind of posterior and inferior, which we can't see in this image, the sinus venosus is locked in as well. So now that we have this bulboventricular loop forming and we started to rotate, we have the resulting position of the heart chambers established that we will see in adulthood. Okay, so now instead of a straight line, where we have truncus arteriosus, bulbus cordis, ventricle, atrium, and sinus venosus, we now have a structure that much more resembles the adult heart. Okay, and we'll see now that we have these chambers in place, because remember this is just one tube, we now need to partition these chambers into right and left halves. So again, looking forward, a preview of what each one of these structures is supposed to develop into. The truncus arteriosus, as we said, is locked in at the most superior poles. It's going to be giving rise to the structures that we see here. So we have the arch of the aorta as well as the pulmonary trunk. Okay, so it'll give rise to the proximal pulmonary trunk as well as the proximal aorta. We look at the bulbus cordis that is directly attached to truncus arteriosus. So we're thinking, what are the next most distal structures? And here we're thinking about the smooth parts of the left and right ventricles. So we're thinking the conus arteriosus, which is here on the right, and then the aortic vestibule, which is over in the left ventricle. The ventricles, of course, are going to give rise to the ventricles. Same with the atrium. Specifically, it's going to give rise to the trabeculated part of the right and left atrium, not the smooth regions. And then the sinus venosus here, we're going to see that it's going to give off, or it's going to develop into the sinus venarum and coronary sinus. And we're going to talk a little bit more about this specific process later, but it's important to recognize that this is where all of the venous inflow is going into the heart. So venous inflow going into the sinus venarum, which will eventually kind of develop into your inferior vena cava, superior vena cava region. Then you're going to get the primitive atrium, so that will develop into this. Then you get the ventricle, and then you get your outflow tracts, and that will be happening on both sides. Okay, so again, we talked about initially you have one big tube that forms the heart, but we know in adult anatomy we have a right and left side. So now what we need to do is we need to partition the atrium and ventricles from one another into right and left halves. So the first thing that's going to happen is we need to form the endocardial cushions. And that's what we're actually seeing happening here in these images. So we have certain components of the developing heart called cardiac jelly. And these cardiac jelly is going to give rise to a ventral cushion and a dorsal cushion. Okay, and this is going to be separating the atrium superiorly and ventricles inferiorly, as well as separating a right side and a left side, or at least starting to form those components. Right, so the anterior and posterior cushions are going to start to come together, and once they fuse, we're going to have canals, and these canals are going to be called the atrioventricular canals. So it goes from right atrium to right ventricle, left atrium to left ventricle. And you can see in this point we actually haven't separated the atrium and ventricles yet, so you have flow going kind of all throughout, but the point being is that the initial stages of partitioning of the atrium and ventricles is forming the cardiac jelly giving rise to the endocardial cushions, those endocardial cushions fusing to create the atrioventricular canals, which allows blood flow to flow from the atrium to the ventricles. Okay, so now that we have the atrioventricular canals formed on the left and right side, we really now need to separate the atrium and ventricles from one another. So the next couple of slides are going to be talking specifically about separation of the atrium into right and left halves. So the first thing that's going to happen, and kind of giving us an idea of what we're looking at here, this view is going to be that coronal view, and then this is going to be that sagittal view. Okay, so this is going to be posterior, this is anterior, superior, inferior, whereas over here we're looking at inferior, superior, and this is right side and left side. Okay, and those are going to be the same for all of the following images that we'll see. So initially, we're going to form a crescent-like structure that's going to rise from the posterior superior wall of the atrium, and it's going to be growing towards the endocardial cushions, which are located here. Okay, this initial structure is called the septum primum. Okay, makes sense. It's going to be the first septum or division that we're forming, so it's going to be called the septum primum, and it's growing towards the cushions. And what we're going to see is as it grows, the space that is slowly starting to close off is going to be called the foramen primum. So if we go through time, we'll see that time has passed, we're starting to close off, this foramen primum has gotten really, really small, and eventually we should completely close that off. So big foramen primum getting smaller and smaller and vanishing. Okay, and this is all through the growth of the septum primum going towards the endocardial cushions located here. We can see the same thing on the transverse view. Initially you have the septum primum forming, it's going to be coming down, and it should close off and fuse with the endocardial cushions. Now, over time, while the septum primum is coming down, a region that's more superior is going to start to degrade. Okay, so this region of the septum primum is going to degrade, and that's going to form a second foramen. This foramen, because it's the second foramen that's forming in the septum primum, is called the foramen secundum. Okay, so the foramen secundum is starting to arise. Over time, we're going to continue to have the septum primum, which is actual membranous structure, collapse down to the endocardial cushions and fuse, but the septum secundum is going to persist. So this is still allowing blood flow to travel from the right side of the heart to the left side of the heart. And we can see that in this image here. This is where both the foramen primum and foramen secundum are present. And again, this is the septum primum, so it has two holes, the foramen primum and foramen secundum. And then over time, that foramen primum should close, and the only thing that's left open is going to be the foramen secundum coming across. Okay. Now, that is pretty much the development of the septum primum. and it forms those two foramen. The next thing that you're going to have is the septum secundum begins to form. So this is the secondary septum. And this is going to be a more membranous, stronger muscular structure. And instead of developing from the posterior superior walls, which we saw with the septum primum, it's going to be forming from the anterior superior walls and growing down. Okay, so this is the septum secundum, and it grows slightly to the right of the first, or the septum primum that was forming. So you can see it starting to form here, developing the septum secundum. And we'll talk a little bit more about how this goes in the next slide. Okay, so as we're moving, the septum secundum, again, is starting to form, and its end goal is going to be to come down and fuse inferiorly. Now, during this process, we're going to start to develop what we're going to call the foramen ovale. So the foramen ovale is a space between the superior and inferior limbs of the septum secundum, so it's this big space here. And we can see if this is the superior limb, this is the inferior limb, the space in between is the foramen ovale. Okay, so blood is able to flow through the foramen ovale and through the septum secundum, which we see here, through the foramen secundum, which we see here, to transmit from the right side of the atrium to the left side of the atrium. Now, over time, the remnants of the septum primum form the valve of the foramen ovale. So what we're going to see here is this structure. Right here, this is what used to be the septum primum, or at this point still is, but the septum primum is going to start to degrade, and then what remains we're going to call the valve of the foramen ovale, because what should happen whenever you're born is the septum primum in this region should close and fuse with the septum secundum, and that's why we call it a valve. Okay, so it's allowing for blood flow through the foramen ovale here, and then the foramen secundum to get from right to left, and eventually when you're born that should close completely. All right, so again, this valve is redirecting blood flow from the right side of the heart to the left side of the heart. At birth, there's a pressure change. We'll talk a little bit about this when we talk about neonatal circulation, but the pressure changes force the left-sided pressures to increase, so it forces the valve to shift to the right, which is going to close it directly against the septum secundum, which should completely close off blood flow from the right and left sides of the atrium. Right, and then what you'll see is this remnant that is going to remain where that fusion occurred. This is going to be called the fossa ovalis. Okay, so the fossa ovalis is the remnant where the foramen ovale used to be. So the foramen ovale is the hole during embryonic circulation, and then when it's closed in adults, you'll be able to see where the foramen secundum is fused with the valve of the foramen ovale. That is going to be called the fossa ovalis. So that's separation of your atrium, but we know we also need to separate the ventricles. So talking about this, there's going to be a couple of different steps that occur. The first is that we're going to have a muscular interventricular septum. So where we had intraatrial septums developing between the atrium, this is going to be interventricular. And so the first one that develops is the muscular interventricular septum. It's growing from the inferior anterior region of the developing heart, and it's going to be growing up towards the endocardial cushions. Okay, so growing towards the endocardial cushions to fuse, and then the resulting space that is going to form before that fusion occurs is going to be called the interventricular foramen. It's just like we saw the atrial foramen in the last slides. Now, the muscular interventricular septum is growing superiorly, but we're also going to have growth from the endocardial cushions. That's going to give rise to a membranous interventricular septum, which is going to grow from superior to inferior. So we have a membranous interventricular septum coming down to meet the muscular septum. Additionally, something that we'll talk about in a couple of more slides is we have the aorticopulmonary septum, which is kind of a separation between the aorta and pulmonary trunk, which is growing from the bulbar ridges. Again, we'll dive into this in a little bit more detail, but that is also going to be a major component that is going to fuse to form the interventricular septum. So when you think about the adult versions of the interventricular septum, there are three components that are contributing. You have the muscular interventricular septum, which is growing from inferior to superior. You have the membranous interventricular septum, which is coming from the endocardial cushions. And then you have the aorticopulmonary septum, which is separating the aorta and pulmonary trunk, which also is one of the attachment points for fusion to form your complete interventricular septum. Okay, so I mentioned we're going to talk about the aorticopulmonary septum. This is where this comes up, and partitioning the outflow tracts. So remember, the outflow tracts are really going to be derived from the truncus arteriosus and bulbus cordis, because the truncus arteriosus is going to give rise to the proximal aorta and the pulmonary trunk, whereas the bulbus cordis is giving those rise to the regions within the ventricles that are then going to go into the aorta and pulmonary trunk. So that's the conus arteriosus and aortic vestibule. So how this initially happens, because again, we have one tube, we need to separate this in half. We're going to have bulbar and truncal ridges form. So these form because neural crest cells are actually going to invade into these regions. So this is neuroectoderm, and they're going to eventually cause midline swellings. They're going to fuse in the midline. Okay, and that fusion is going to result in this yellow structure we see here, and that's going to be called the aorticopulmonary septum. Now, initially, this is a direct sheet of tissue that is just in the midline, and that's separating the pulmonary trunk from the aorta. But what we're also going to have as development continues is we're going to have a 180-degree spiral that is going to result in the adult orientation of the outflow tracts. So when we think about it, initially the pulmonary trunk is coming from the right ventricle. But if you look in the adult anatomy over here, so you have the pulmonary trunk on the right, it's going to come off the heart on the left. And the aorta is the opposite. The aorta is coming from the left ventricle, but it comes off the heart on the right side. So at some point we need the spiral in order to get this adult anatomy, and that's what's happening here. So that 180-degree spiraling resulting in adult orientation of the outflow tracts. Okay, and so thinking about the derivatives, we have the bulbus cordis on the right side giving rise to the conus arteriosus. That's that smooth region of the heart just before you transition from the ventricle into the pulmonary trunk. On the left side, the bulbus cordis is giving rise to the aortic vestibule. Again, that smooth region just before you enter into the aorta. And then the truncus arteriosus, really those proximal outflow tracts, giving rise on the right to the pulmonary trunk, and on the left giving rise to the proximal aorta. Okay, so again, partitioning the outflow tracts, we have the bulbar and truncal ridges forming neuroectoderm invasion, forming a flat sheet that separates into right and left sides. We then need to have a 180-degree spiral in order to get the adult orientation that we would expect for the outflow tracts. Okay, I know this is a lot of information, so we're going to go through a quick summary of what we've done up to this point. So initially in developing the heart, we had the angioblastic cords forming. Okay, those angioblastic cords form, they then canalize to form the endocardial heart tubes. Those heart tubes fuse at the region of the primordium heart, and specifically giving rise to the endocardium. Okay, the myocardium epicardium, we didn't talk about this, but this is going to be derived from the splanchnic mesoderm associated. Okay, so we're just specifically talking about endocardium throughout this development. Now, once the heart tubes fuse, we're then going to get primitive dilations. And so we have the truncus arteriosus, bulbus cordis, ventricle, atrium, sinus venosus from a superior to inferior standpoint. Because the truncus arteriosus and sinus venosus are locked into place because of their attachments, and there's differential growth between the bulbus cordis and ventricle, we get the bulboventricular loop. Okay, this is going to allow for the ventricle to shift to the left, the bulbus cordis to shift to the right, and then the atrium is going to shift posterior to the developing heart. And this is going to be the orientation that we're expecting for adult anatomy. Okay, so now we have the relative relationships of the heart chambers formed. Again, the venous return is going to be from the sinus venosus and atrium. We expect this is how blood flow returns to the heart in adults. And the outflow tract via the truncus arteriosus and ventricles. Again, recognizing this is eventually going to give rise to your aorta and pulmonary trunk. Now, again, this is only one tube, so that the fusion of the heart tubes gave us one tube, but we need to partition these into left and right sides. Okay, we do this by partitioning the atria. The atria is partitioned initially by the endocardial cushions forming, which is going to give rise to our atrioventricular canals. Then we have the septum primum that's growing from the superior posterior wall. The foramen that is present initially is going to be the foramen primum. Apoptosis at the superior pole is going to give rise to the foramen secundum. So recognize that in atrial partitioning, both foramen primum, and foramen secundum are holes associated with the septum primum. But later, the septum secundum forms directly to the right of the septum primum, and then this is going to be leaving a space called the foramen ovale. This is allowing blood to transit from the right heart to the left heart, and it goes through the foramen ovale and then through the foramen secundum. Okay, eventually that should fuse completely to give us the intraatrial septum. We also have to separate the ventricles. This is going to start with the muscular interventricular septum, which is going to be rising from the floor, going up to the endocardial cushions, and it's met by the membranous interventricular septum, which is rising at the endocardial cushions and coming down. Again, we also have the aorticopulmonary septum, which is a component here. And partitioning the outflow tracts, we had those bulbar and truncal ridges, again, neural crest cell migration, causing that, so this is neuroectoderm. These ridges are giving rise to the aorticopulmonary septum, but again, we have to have that 180-degree rotation because we know that the aorta is going to come off on the right side of the heart, even though it's coming from the left ventricle, and then the pulmonary trunk is going to come off of the left side of the heart, even though it's coming from the right ventricle. Okay, so we had that 180-degree rotation. And that takes us through a summary of development of the heart up to this point. Okay, so now to dive in a little bit more about what happens to the sinus venosus in development. So initially, the sinus venosus is receiving the venous return from the entire body. And there's going to be a couple of different components that we want to point out here. So it's attached to the atrium, but we initially have a right horn and a left horn. Okay, what you'll see here for each horn, you're going to have a superior and inferior cardinal vein. These cardinal veins are receiving blood from the embryo. So superior cardinal vein, inferior cardinal vein makes sense. With something above, something below the heart. The vitelline veins, these are receiving blood from the umbilical vesicle. Okay, so you're going to have one on the right and one on the left. And then we're also going to have a umbilical vein, which is coming from the placenta. Okay, so these are all the components of your right and left sinus horns. So again, initially you're going to have one on either side with each component. But eventually what's going to happen is we're going to see a shift towards right-sided venous return. And this makes sense to us because in the adults, most of the blood flow is going to be coming through the right atrium, through the superior vena cava and inferior vena cava. So we know that venous return has to shift to the right as opposed to being directly in midline. Right. So what's going to happen is on the left side, you're actually going to have degradation of the umbilical and vitelline veins. So these are going to degrade. Initially, on top of this, the left superior cardinal vein is actually going to anastomose with the right cardinal vein. And we'll see this in a moment, but that's kind of giving off that brachiocephalic vein. And we'll, again, see this more in more detail in another image. So eventually, what we'll actually see is the left horn is going to really degrade dramatically, and its final contribution to the adult heart is going to be the coronary sinus. And if you recall, the coronary sinus is what is returning blood flow to the heart from the heart itself. Okay, so the veins run with the coronary arteries. Those veins drain into the coronary sinus, and the coronary sinus opens up into the right atrium, returning blood flow from the heart. The right horn, however, is going to be it's going to persist as really the majority of the venous return for the body. So part of it's going to be incorporated into the right atrium. Specifically, those smooth regions in the posterior wall are going to be contributed to the sinus venosus, not the atrium itself. And then the crista terminalis is kind of a ridge that separates the smooth region and the trabecular portion of the right atrium, and that's where that SA node is going to be housed. But again, that right horn is going to develop in the superior vena cava, inferior vena cava, and then the smooth part of the right atrium. Around the same time, we know that the left atrium also needs to receive blood from the lungs. So we're going to start to see those primordial pulmonary veins from the left atrium extending out to the developing lungs, so that way you can receive blood flow from the lungs as well. So this is another image that's kind of showing the degradation of the left sinus horn over time until we get to the adult structures. And so we can get our orientation back, we're looking from the posterior region of the heart to anterior. So we're looking from the back. So what we can see here is initially you have the right sinus horn, left sinus horn. You have the cardinal veins on each side. You have the vitelline veins and the umbilical veins. Everything is symmetric. Over time, what's going to happen is the umbilical and vitelline veins on the left are going to degrade. You're starting to get some of this anterior or superior cardinal vein degrading as well on the left, because we're going to start to anastomose on the left side with the right side. So you start to see this anastomose forming, and then when it does form, that is going to eventually give rise to the brachiocephalic vein like we see in the adults. Okay. Then on the right, everything is really persisting. We have the cardinal vein superiorly, we have the cardinal vein is going to persist, and then it's eventually going to turn into the superior vena cava and inferior vena cava, whereas really this left horn over here is starting to wrap around the heart itself, and that's eventually going to form your coronary sinus. Okay, so there goes your initial developments for the heart. So we can see in the adult, really the right horn is persisting, giving off the majority of the structures, whereas the left horn is primarily degrading. Okay, so that's talking about the inflow tracts, so getting blood to the heart, but we also have to talk about blood getting away from the heart. And this is where we have to recall a little bit about our pharyngeal apparatus. So not giving you too much PTSD, but just briefly thinking about this. We talked initially about each one of these pharyngeal arches giving rise to musculature, bones, ligaments, and being associated with a nerve. It's also important to know each one of these pharyngeal arches is associated with a vessel. Okay, and we're going to talk now about how vessels are initially going to give rise to different components in the adult anatomy, and just keeping up which pharyngeal arch is corresponding with each structure. Okay, so here we're talking about pharyngeal arch derivatives. I think it's important to recognize this is kind of what we're looking at initially in development, that the pharyngeal arches or pharyngeal arteries are originally going to form from an aortic sac. So it's just one continuous structure. Eventually, the arteries are going to branch off, and then they're going to develop into their adult structures. There are 6 total pairs, just like there are 6 total pharyngeal arches, but it's important to know for the pharyngeal arch arteries that 1, 2, and 5 degrade pretty significantly. So 1 and 2 are going to have small components in the adult anatomy. 5 is going to be completely absent. Okay, so the main vessels that you're going to keep up with are the branches from pharyngeal arch 3, pharyngeal arch 4, and pharyngeal arch 6. So as we look at this, we're going to start with pharyngeal arch number 1. Remember, pharyngeal arch number 1 gave off a lot of things that started with an M, and that's going to be the same case here. We're going to form part of the maxillary artery. Okay, so the first pharyngeal arch giving rise to components of the maxillary artery. The second pharyngeal arch gives rise to parts of the stapedial artery. Okay, again, these are up in the skull region. So maxillary artery for 1, stapedial artery for 2. Now is where we're starting to see things down in the region of your great vessels. So pharyngeal arch 3 is going to give off the common carotid and proximal internal carotid on each side. And you can see this indicated here. So 3, pharyngeal arch 3, giving rise to the common carotids, as well as part of the internal carotid on either side. This is on both the right and the left. Okay. For pharyngeal arch 4, this is where we're going to start to see a little bit of differentiation between the left side and the right side. So on the left side, it's going to give the region between the left common and subclavian veins. So over here, we have the left common carotid, we have the left subclavian, and that region of the aortic arch, just in between, that's going to be derived from the 4th pharyngeal arch. So the 4th pharyngeal artery, right there. On the right side, it's going to give the region that is just that proximal subclavian. Okay, I know it's a little bit difficult to see here, but in pink, you have the brachiocephalic vein, and then it's going to give off the right subclavian. That proximal region of that right subclavian is going to be derived from the 4th pharyngeal arch. Okay, and then pharyngeal arch number 6, giving rise, the arteries that it's going to give rise to is on the left side, you have the proximal left pulmonary artery. Okay, so proximal left pulmonary artery coming up, and then also remember, this is something that we'll talk about in a little bit more detail. There is going to be a connection between the pulmonary trunk here and the descending aorta. This is going to be known as the ductus arteriosus. It's going to be really important for neonatal and fetal blood supply, but this is being derived from the 6th pharyngeal arch. Okay, and then on the right side, we're going to have just that right proximal pulmonary artery. Okay, so going through this one more time really quickly, the big pharyngeal arches that you need to keep up with contributing to adult vasculature is going to be pharyngeal arches 3, 4, and 6. 3 is contributing to the common carotid bilaterally and the proximal internal carotid arteries. We can see that in orange here. The pharyngeal arch number 4, the region between the left common carotid and left subclavian, so it's forming part of that aortic arch. On the right side, it's just forming part of that right proximal subclavian. And then for pharyngeal arch number 6, we have the right proximal pulmonary artery, left proximal pulmonary artery, and then again on the left side we have the ductus arteriosus, which is connecting the pulmonary trunk and the descending aorta. Now that we've talked about the blood flow that is going to be disseminated from the superior poles of the aorta, we need to talk about the development of the dorsal aortic branches. Okay, and the main thing here is if we can think about the adult anatomy, then we'll be really well positioned to understand the embryonic components. So 3 major components that we're going to talk about: ventral branches, lateral branches, and dorsal branches. The ventral components are going to be broken down into 3 other categories. We're going to have the vitelline components, the proximal umbilical, and distal umbilical. So these are components that we're going to be thinking about. But really, if you think to the adult anatomy, it makes sense. There are a couple of ventral branches that come off the abdominal aorta. So the vitelline components are corresponding to anything that's supplying the GI system. And we think, what are the 3 major vessels that come off the abdominal aorta right off the front that supply the GI system? You have the celiac trunk, you have the superior mesenteric artery, and the inferior mesenteric artery. Okay, we're also more proximal than seen in this image is you're going to have the esophageal branches, but really supplying the GI system. So celiac trunk, SMA, IMA, esophageal branches. Those are all from the vitelline components off of the ventral branches of the aorta. Lateral branches, we can actually see those just in the image if we remember our adult anatomy. We have suprarenal arteries, so we have superior suprarenal, middle suprarenal, inferior suprarenal. We have renal arteries, and then we have the gonadal arteries. And again, the gonadal arteries can be either testicular or ovarian depending on the sex of the patient. Okay, so the lateral branches are going to be the renal, suprarenal, gonadal. Now, we do need to keep track of the proximal umbilical and distal umbilical. These are coming off the anterior aspects of the aorta, but that's happening more distally in the pelvis. So that's after the common iliac splits into an internal iliac and an external iliac. So coming off that internal iliac, we're going to have the umbilical artery, and that umbilical artery is going to go off the branches superior vesicular and the umbilical. Okay, now distally, that umbilical artery is going to degrade in adults because there's no reason for us to be sending blood through our umbilical artery out of our belly button. So it's going to degrade and it's going to become the medial umbilical ligaments. But these are all the ventral branches. So running through this one more time, there are 3 major components we're going to keep track of: ventral, lateral, and dorsal branches. The ventral branches we are broken down into vitelline, proximal, and distal components. The vitelline components is everything that's supplying the GI system. So you have the esophageal branches, your celiac trunk, superior mesenteric artery, inferior mesenteric artery. The proximal umbilical vessel is going to give rise to the superior vesicular vessel in adults, the umbilical artery, and the internal iliac, whereas the distal regions are just going to contribute to the medial umbilical ligaments. Coming off laterally off the abdominal aorta, you're going to have your suprarenal arteries, you're going to have your renal artery, and you're going to have your gonadal arteries. Okay, all other branches of the abdominal aorta, the thoracic branches that are in the thorax, the lumbar branches that are in the region of the abdominal aorta, the sacral branches that are more inferior, these are all going to be components of the dorsal branches. Okay, so again, you have ventral branches, lateral branches, dorsal branches. If you remember the adult anatomy, it's the exact same way as it will be in development. Okay, so fetal circulation is very, very important in order to understand the actual clinical correlations at the end of this video. So we're going to spend a little bit of time here. So fetal circulation pathway, this is once we've started to develop the heart in its entirety. So we have the right atrium, right ventricle, left atrium, left ventricle. Now, at this time, remember that the foramen ovale is still open. So we're allowing blood to flow from the right atrium to the left atrium through that foramen ovale. The foramen ovale does not close until you're born and the pressures from the left side of the heart increase, forcing it shut. So during this fetal circulation, we're going to start with the placenta because this is where oxygenated blood is coming from mom. So we start at the placenta and it's going to flow towards the fetus through the umbilical vein. Okay, this umbilical vein giving oxygenated blood. Normally the umbilical vein would come through and there's a possibility for it to go to the liver, but we really want to bypass the liver because this has already been filtered by mom. We want to get this to the baby as fast as possible. So we're going to bypass the liver via what we're going to call the ductus venosus. So the ductus venosus is, again, a pathway that allows us to bypass the liver and go directly into the inferior vena cava. Okay, so we're bypassing the liver in the portal system. So we get into the inferior vena cava. So now we can see that we've got some oxygenated blood going into the heart. That oxygenated blood is then going to go into the right atrium. Okay, again, because the fetus is in mom, there is no respiration happening. The developing fetus is not breathing. There's no way to oxygenate blood itself. So there's no reason for us to send blood to the right ventricle and into the pulmonary circulation. Because if we go into the pulmonary circulation, again, there's not going to be any additional oxygenation happening. So really we want to bypass the right side of this heart completely. So we go from the right atrium into the left atrium. Go from the left atrium into the left ventricle. And again, because we're still got oxygenated blood because it's coming from mom, we go from the left ventricle directly into the aorta. Okay, and then that's going to distribute across the systemic circulation. As we come down, we're going to go through the dorsal aorta, or the descending aorta, and then we're going to have those umbilical arteries that are going to return the blood flow to mom, so that way mom can, reoxygenate and send it back to us through the umbilical vein. And what I want to point out, I said that we want to bypass the pulmonary system, but we can't just bypass it completely because we do have the tricuspid valve, so that's allowing some blood flow from the right atrium into the right ventricle. So what do we do with this blood when it's going in this region? Well, we're going to send blood from the right ventricle towards the lungs, but again, we said we don't want it to go all the way to the lungs if we can prevent it. So what's going to happen is we're going to have a canal here called the ductus arteriosus. And again, we mentioned this arising from the sixth pharyngeal arch, and it's connecting the pulmonary trunk and pulmonary arteries to the descending aorta. And this is allowing blood flow to flow through from the pulmonary trunk to the aorta because of the pressure gradients in neonates. Okay, so going through one more time the different components, we have the umbilical artery coming from, umbilical vein, one umbilical vein coming from mom. We're bypassing the liver, the ductus venosus. That's going to allow blood flow to get into the inferior vena cava. We're then going to get into the right atrium. The majority of blood flow is going to go into the left atrium, to the left ventricle, to the aorta. Some blood flow will get into the right ventricle, which will then send it to the lungs. Again, because the lungs cannot oxygenate anything before you're born, there's no reason for blood to be here. So we have the ductus arteriosus, which is allowing us to shunt blood from the pulmonary trunk into the aorta. So once all the blood is in the aorta, it can distribute across throughout the body, and then in order to return blood back to mom, so that way it can be reoxygenated and sent back to us, we're going to give it to mom through the umbilical arteries, which are going to go down to the placenta, which is that interface between maternal and fetal circulation. Now, in thinking about what some of these embryonic developments are and what their derivatives are going to be, let's start with the left umbilical vein, because again, the umbilical vein should degrade in adults because there is no additional blood flow coming from the placenta. That gets clipped when you're born, so there's no reason for it. So what this is going to develop into is a ligamentum teres hepatis. So this is an actual ligament on the bottom. So you can see that it goes to the liver. It's a ligament on the bottom of the falciform, which is a remnant of the umbilical vein. So you'll be able to see this on the cadavers. The ductus venosus, again, this is a pathway to allow blood flow to shunt from the placental circulation directly into the inferior vena cava, bypassing the liver. You don't have any blood flowing through it in adults, so it's just going to degrade, and it's going to give rise to the ligamentum venosum. We talked about the foramen ovale. It is going to close as soon as increased blood pressures occur in the left atrium. Okay, closing it completely, that will give rise to the fossa ovalis in the adult anatomy. The ductus arteriosus, we said because we don't want blood going to the lungs, we're going to shunt it into the aorta. In the adult, we do not want this to be open. We want to close this. We want separate flow, so that way blood is going to the lungs, being oxygenated, and then coming into the left atrium. So we need to close this in adults, and when it's closed, it's going to be called the ligamentum arteriosum. And lastly, these umbilical arteries, because there's no blood that needs to be flowing through our belly button back to mom, we're going to say, hey, we need to close these down, and then they're going to fuse, and then they're going to become the medial umbilical ligaments. And remember, this is primarily that distal region of the umbilical arteries. Okay, so now we begin to breathe. We get neonatal circulation. What changes? So as soon as we start to breathe, that left atrial pressure is going to increase because we get increased pulmonary blood flow going to the lungs. So as that left atrial pressure increases, it's going to force that valve of the foramen ovale shut. So it forces it to the left, and it's going to close completely, or at least it should close completely. So now the blood is flowing from the right atrium to the right ventricle to the lungs. So we go right atrium, right ventricle to the lungs. It gets oxygenated by the baby. Baby brings blood back through the pulmonary veins into the left atrium, going to the left ventricle, into the aorta, disseminating across the body. Okay. Now the ductus arteriosus should begin to close, because remember, we don't want that to be open in adults or in developing babies, because what's going to happen there is the aorta is going to be a really high pressure system. It will actually force blood back into the pulmonary trunk, away from where we need it to go. Okay, so this is mediated by bradykinin, so that's the chemical mediator to allow for closure of the ductus arteriosus. Okay, now we're going to dive into some clinical anatomy. So the first thing that we're going to talk about is alterations in heart formation, and we need to define a couple of terms that we're going to use throughout the next couple of clinical cases to make sure we're on the same page. The first is a left to right shunt. So a left to right shunt means that blood is flowing from the left side of the heart, which should be oxygenated, to the right side of the heart, which is deoxygenated blood flow. And so over here on the right, we can see what the normal pressures and oxygenation of the heart chambers are. So you can see in the right atrium, the pressures are low, 0 to 6 millimeters mercury, and the oxygenation status is low as well, about 70%. We get into the right ventricle, it's going to increase a little bit, the pressures, and that makes sense because we're going to be pumping it into the pulmonary trunk, but it's still oxygenation low. Pulmonary trunk, oxygenation is low, haven't reached the lungs, haven't been able to oxygenate it yet, and the pressure should mirror that of the ventricle. Once I get into the right atrium, however, left atrium, now I have increased oxygenation, greater than 95% to 100%. Still low pressure, but I've got oxygenation. Get into the left ventricle, still fully oxygenated, shouldn't have any mixture of blood, and now the pressures are going to increase because I'm going to have to force this blood into the aorta. Okay, so it helps to keep in mind the general pressure gradients as well as oxygenation when we're talking about these different cardiac abnormalities. So when we have a left to right shunting of blood, that means that we have blood from the left side over here shunting to the right. This is going to be oxygenated blood mixing with deoxygenated blood, but again, because it's going towards the left, there's no mixture getting into the aorta. So this is going to be an acyanotic heart defect, meaning it's not going to cause decreased oxygenation in the systemic circulation. A right to left shunt, however, will do the opposite. So right to left shunt is allowing blood from the right side of the heart, which is deoxygenated, to mix with the left side chambers before it is distributed through the systemic circulation through the aorta. So this is going to cause cyanotic heart defects, meaning we have deoxygenated blood in the systemic system. So we think left to right shunts, acyanotic, right to left shunts, cyanotic heart defects. Okay, and again, each chamber has an average pressure and oxygenation. If you know what the defects are, a lot of these defects, we're going to talk about defects in the septum or defects in structures failing to regress from embryonic derivatives. You should be able to predict how these changes are going to occur. So general principle is flow is going to be from a high pressured area to a low pressured area. So if there's for instance, if there's a hole in between these ventricles, where do you think blood is going to flow? The high pressured side to the low pressure side. And if it's flowing from the left side of the heart to the right side of the heart, we said this is a left to right shunt, it will be acyanotic. Okay, so we're going to start with our acyanotic heart defects, and we're going to move on to our cyanotic heart defects. Starting with our first acyanotic heart defect, we're going to talk about an atrial septal defect. Now, atrial septal defects in general just refer to a defect of the atrial septum, okay, but this can happen for a variety of reasons. So one reason is we could have what's called a POD, patent foramen ovale. This is an incomplete fusion of the foramen ovale valve and the septum secundum. So it's not a big, large open defect. It's usually small, doesn't have a whole lot of clinical significance other than it's just an incidental finding that you'll occasionally find. Okay, the ostium septum defects are a little bit more important to recognize. So this is abnormal reabsorption of the septum primum or development of the septum secundum, and this results in a patent foramen ovale. So the difference here is patent foramen ovale versus probe patent foramen. Probe patent means it's really small. Patent foramen ovale is going to be a larger defect, and this can happen at different regions. So it can happen down the region where you had the septum or the foramen primum, or it can happen in the region where you had the septum secundum or foramen secundum. The ostium primum defects, this is going to be an issue with the endocardial cushions or atrioventricular septum. So this is going to be happening more at the fusion point, and this can lead to patent foramen primum, which is where you have that failure to fuse with the endocardial cushions. Okay, so this again, the septum primum, remember, is coming down, fusing with the endocardial cushions. When it fuses, the foramen primum should completely go away. But if that does not happen, the endocardial cushions don't form, the atrioventricular septum doesn't form correctly, then you're going to get a patent foramen primum. Okay, so these are just different flavors of the same thing. An atrial septal defect, allowing blood flow to transmit between the right atrium and left atrium. In this case, it's acyanotic because blood is going to start in the left atrium, which is slightly higher pressure than the right atrium, and it's going to shift the blood back into the right atrium. So you're not getting any deoxygenated blood in the systemic circulation, which is what makes this an acyanotic heart defect. Next, we talk about ventricular septal defects. We kind of mentioned this a little bit already, but this ventricular septal defect is any defect along the interventricular septum that is allowing blood to flow between the ventricles. Okay, in this case, this is going to result in shunting of blood from the left side to the right side of the heart, which makes it an acyanotic defect. And this is because the pressure in the left ventricle is greater than that in the right ventricle. Okay, this is due to failure of formation of the membranous or muscular interventricular septum. Okay, and so the big thing here is we're getting that right to left shunting of blood over time. And what this image is showing is as you get into adulthood, this right ventricle has a lot more blood flow going into it than it's used to. So it's going to start to hypertrophy. It's going to get stronger and stronger, and eventually it's going to have the same pressure or the ability to apply the same pressure as the left side of the heart. And when this happens, you're now going to get shunting of blood from the right side of the heart to the left side of the heart, and this process is called Eisenmenger syndrome. Again, this is just a fun little clinical point. The big thing you want to point out is initially, after birth, you're going to get left to right shunting of blood because the pressure on the left side of the heart is greater than that on the right. Next up, we have the persistent ductus arteriosus. So again, we talked about the ductus arteriosus connecting the pulmonary system with the aorta, and this makes sense in development, but it doesn't make sense once you're born. So this is failure of the ductus arteriosus to close, and again, that should be happening under the direction of bradykinin. When this doesn't happen, we're going to get left to right shunting, and the reason for this is because we're going to get the aorta, which is a high pressure system, sending blood into the pulmonary trunk. Okay, so it sends blood into the pulmonary trunk here, so it's not getting out into circulation, but now you've got this oxygenated blood going to the lungs. It's really high pressured. Eventually what this can cause is it can cause hypertension in your pulmonary system, which puts a lot of strain on your right ventricle. When that right ventricle is under a lot of strain, it can actually lead to heart failure, where blood is backing up into the right system and then into the systemic circulation. Okay, so one of the associations that you need to make here is that PDA, or ductus arteriosus, is associated with maternal rubella infection. Okay, but again, this is because you have failure of that ductus arteriosus to degrade from that embryonic stage. All right, next up, and this will be our last acyanotic defect, we're going to talk about coarctation of the aorta. Coarctation of the aorta refers to stenosis or narrowing of the aorta, and this is commonly going to happen near the junction of the ductus arteriosus. So it can happen preductal or postductal, which is going to give different clinical signs. But if it's before the left subclavian vein, so this is before the left subclavian, which would be this image here, you're going to get high blood pressure in the right upper extremities, and then you're going to get low blood pressure distally. You also have low blood pressure in the left upper extremity because, again, the stenosis happens before the subclavian, which is going to feed the left upper extremity. If the stenosis happens after the left subclavian, so now you've got blood flowing through the aorta, it's going to the upper limbs, it's going to the head, but now it can't go down to the lower limbs. And that's where we can get low blood pressure in the lower limbs, high blood pressure in the upper limbs. So this is one of those, this is an instance in which you check blood pressure in newborns in both the upper limb and lower limb. All right, so here's a summary of our acyanotic defects, the big conditions, key features, how blood flow is going to change, and associations you might want to know. So atrial septal defects, again, key feature, you have a hole between the atria. This is going to allow left to right shunting via the defect, so you're not getting decreased oxygenation and systemic circulation associated with Downs and maternal alcohol use. A VSD is, again, just a hole in between the ventricles, allowing shunting of blood from the left side to the right, also associated with Downs and maternal alcohol use. The PDA, this is where the ductus arteriosus is remaining opening. This is connecting the pulmonary trunk and the aorta. This allows left to right shunting from the aorta, which is the high pressure system, into the pulmonary trunk, which is the low pressure system. And then this is going to be associated with maternal rubella infections primarily. Down syndrome, partially, I would really key in on that maternal rubella syndrome. Lastly, we have coarctation of the aorta. This is narrowing of the aorta. Usually it's distal to the subclavian, but it could be slightly proximal. But again, really narrowing of the aorta, and this is going to impede blood flow to the lower body while increasing the pressures to the upper body where blood is able to flow, and this is associated with Turner syndrome. First cyanotic defect we're going to talk about is tricuspid atresia. And what I want to talk about with the cyanotic defects, these are much worse prognostically because this means you're getting deoxygenated blood into the systemic circulation. So we know that's going to be the end result. So now we're just going to have to figure out why that's the case. So in tricuspid atresia, we do not have the tricuspid valve forming. So tricuspid valve does not form between the right atrium and right ventricle. So we do not get blood flow here. So in order to compensate, your body is going to do a couple of things in order to make sure that we still have blood flow occurring. So one is we're going to keep the atrial septal, we're going to have an atrial septal defect, allowing blood flow to go from the right side of the heart to the left side of the heart, because there's no way for us to get into the right ventricle otherwise. And it's important that we still have blood getting into the right ventricle, because going to the right ventricle is the only way that we can get blood to the lungs, which is going to allow it to be oxygenated so that we can get oxygenated blood flowing throughout the body. So we need to get it to the right ventricle, so we're going to have an ASD, allowing it to go from right atrium to left atrium. We're going to allow it to go into the left ventricle, but then we're also going to have a ventricular septal defect, allowing for blood to flow into the right ventricle. Okay, so we failed to develop the tricuspid valve. We have a ASD and a VSD. And because we're going to have mixing of blood here, so this is deoxygenated blood going from the right side of the heart to the left side of the heart. This is going to be deoxygenated blood in general. Again, the pulmonary veins that are receiving oxygenated blood are also mixing here. So you get mixed blood, so it's deoxygenated, going into the aorta and going into the pulmonary trunk. So overall, the end result is deoxygenated blood systemically. Next up, we have persistent truncus arteriosus. So remember that the truncus arteriosus is giving rise to those outflow vessels, and we're supposed to have the aorticopulmonary septum separating the pulmonary trunk and aorta. Well, if that does not happen, and we have failed aorticopulmonary septum formation, we're going to have just one giant tube that's leading into both the pulmonary trunk and the aorta. So when this happens, we're getting mixture of blood coming from the coming over here from the left ventricle and right ventricle, going into one giant tube, mixing, so we have decreased oxygenation going to the systemic circulation. So that's your persistent truncus arteriosus. Next up, one that's really scary is transposition of the great vessels. This happens when you have failure of that aorticopulmonary septum to do its 180-degree rotation. So what happens is that the right ventricle is actually connected to the aorta, and the left ventricle is connected to the pulmonary trunk. So when this happens, you end up with two completely separate circulations. You have blood coming from the baby going into the inferior vena cava, superior vena cava, getting into the right atrium, goes into the right ventricle, then it goes directly back into the aorta, never touching the lungs, never getting oxygenated, and just goes back into circulation. So there's no way to oxygenate the blood that's supplying the body. And then over here, we just have constant oxygenation without it doing anything. So let's say we start in the left atrium, goes to the left ventricle, then we're immediately going to the pulmonary trunk, which goes to the lungs. Okay, great. We're oxygenating the lungs, or we're oxygenating the blood from the lungs. It's coming back, and it goes directly back into the left atrium. It doesn't go to systemic circulation, does not supply oxygen to the rest of the body. So this is going to be a cyanotic defect because we've got deoxygenated blood in the systemic circulation. Okay, and one more time, transposition of the great vessels occurs because the aorticopulmonary septum fails to perform its 180-degree spiral. All right, next up we have tetralogy of Fallot. Fun thing here, tetralogy is going to represent the 4 different defects that are associated. So the first defect is pulmonary stenosis. So this means that the pulmonary valve is going to be narrowed, so it's going to be hard for blood to be forced through. And this is really the one that I think about starting everything, and everything else falls off after it. And what I mean by that is because we get pulmonary stenosis, it's really hard to force blood through. The right ventricle is going to start to get larger in hypertrophy. It's going to grow stronger. As this occurs, it's going to force the interventricular septum to fail to fuse correctly, and we get a VSD, so ventricular septal defect. At the same time, we're also going to get what we're going to call an overriding aorta. And an overriding aorta just means that the aorta is going to shift a little bit towards the right, and so that way, and what's going to happen because of that is blood flow from the right ventricle is going to be able to go directly into the aorta. So, over time, as the right ventricle hypertrophies, it grows stronger, forces blood to the left side of the heart. So we're getting mixing of blood. The deoxygenated blood mixes with the oxygenated blood on the left side of the heart, and then is sent into systemic circulation So now we have the end result of deoxygenated blood going systemically. So for tetralogy flow, 4 defects you need to keep in mind. You have a pulmonary stenosis, right ventricular hypertrophy, VSD, and overriding aorta. And the end result here is going to result in deoxygenated blood going into systemic circulation. All right, so the cyanotic heart defects. One way to remember this is remember the terrible T's. You have tricuspid atresia, truncus arteriosus, transposition of the great arteries, and tetralogy of flow. Again, the key features for each of these are important to recognize as well as their impact on blood flow. So key features of tricuspid atresia, you have complete occlusion of the right atrioventricular canal, meaning that the tricuspid valve does not form. That is going to lead to an underdeveloped right ventricle, an atrial septal defect, and ventricular septal defect. You can see that down here at the bottom. Truncus arteriosus, this is going to be failure of the aortopulmonary septum to form. It's often associated with a VSD, and this is going to allow for deoxygenated blood to go directly into the aortic and pulmonary trunk. So we got that mixing happening, and that's going to allow for deoxygenated blood to reach systemic circulation. Tetralogy of flow, transposition of the great arteries. Again, this is really due to the failure of the aortopulmonary septal spiral, and this is allowing the right ventricle to connect to the aorta and the left ventricle to connect to the pulmonary trunk. So we have 2 separate circulatory processes, but neither one of these is allowing for oxygenation to get to the systemic circulation. And then lastly, tetralogy of flow associated with 4 defects. Remember, you have pulmonary stenosis, right ventricular hypertrophy, VSD, overriding aorta. And all of this is going to, again, because cyanotic heart defects, all of these are going to lead to deoxygenated blood reaching systemic circulation. And with that should bring us to an end of this